Good day, class. This is Ma'am Renz again, and welcome to Science Time. We are now on 4th quarter, Module 2, Cells at Work, Mitosis, and Meiosis. For our most essential learning competencies, we have compare mitosis and meiosis and their role in the cell division cycle, explain the significance of meiosis in maintaining the chromosome number, and for the following objectives, letter A, discuss the cell cycle and the changes that occurs during mitosis and meiosis. Recognize the significance of meiosis in maintaining the chromosome number and illustrate two graphic organizers the process of gametogenesis. Now, where did all it started? Yung topic natin today. So, dito yan nagsimula sa cell. Now, remember, there are three parts or three main parts of a cell. Namely, the jelly part of the cell, which is the cytoplasm, the outer covering called the cell membrane, and the center of it all is the nucleus, which is the brain of the cell. Inside the nucleus, we have what we call the chromosomes, in which these chromosomes have DNA inside, and that is called deoxyribonucleic acid. Inside the DNA are genes, which codes for the DNA for instructions, which is unique for each type of living creatures. So these are the sample pictures of a cell and their chromosomes. Chromosomes yung mga XX. Now, inside the chromosomes are the DNA packed with protein. And inside the DNA are the genes, which give us a different traits for heredity and variation. So when we say chromosomes, is either X or Y chromosomes. Chromosomes ensures DNA or deoxyribonucleic acid is accurately copied and distributed during cell division. So dapat tama yung pagkopya, walang mali, at tama din yung pagdidistribute kapag hinati na yung cell. Okay? It's very important. Later, we will discuss it. So we have to unlock first some difficulties. Number 1, it is a part of the cell cycle when replicated chromosomes are separated into two new nuclei. Nuclei is the plural for nucleus. So this is called mitosis. Part siya ng cell cycle. Next, number 2, it is a specialized type of cell division that reduces the number or the chromosome number by half, creating 4 haploid cells, each genetically distinct from the parent cell that gave rise to them. So this is meiosis. Yung mitosis kasi identical, tapos dalawa yung na-produce na daughter cell or 2 nuclei. Dito naman sa meiosis, 4 haploid cells yung nabubuo, and yung 4 na yun magkakaiba. Okay, magkakaiba sila, hindi sila identical. Third, it is a repeated drama of cell division, and that is called the cell cycle. Kaya nga repeated cycle. Next, okay, cell division is the process by which a parent cell divides into 2 or more daughter cells. So once na nahati yung isang cell, cell division yun. It usually occurs as part of a larger cell cycle. So part lang siya ng cell cycle. Next is the quality of a cell or organism having a single set of chromosomes. It's called haploid. Half. Okay, half yung ibig sabihin. From example, 46 chromosomes, nag-half siya, 23. Yun yung single set, yung 23 na yun. Usually, haploid is for meiosis. Next, diploid is the quality of cell or organism having 2 sets of chromosomes. So, yung isang set is 23. So, kung 2 sets siya, 46 chromosomes. Sa human chromosomes yung tinutukoy ko. So, that is diploid. Usually, it is for mitosis. Next, also known as sex cells, either sperm or egg, ang tawag doon gametes. Next, 8 is the origin and development of the sperm cells within the male reproductive organs. It's called spermatogenesis. And 9 is the origin and development of the egg cells within the female reproductive organs. It's called oogenesis. Okay, for female. Now, let's take a look on the cell cycle. So this is a cell cycle. As I've said, cell division is only a portion of the cell cycle. Cell cycle is composed of interphase and M phase. M phase stands for either mitosis phase or meiosis phase, depending on the type of cell. If it is mitosis, then it is all the body parts except the sex cells. Okay? So, lahat maliban sa egg and sperm. But if it is meiosis, excluded, hindi kasama ang body cells, specialized lang siya sa egg and sperm. That is meiosis. Okay? So now, during what stage of the cell cycle does G1S and G2 phase happen? G1 stands for growth 1. S stands for synthesis. G2, growth 2. So, during what stage? It's interphase. Next, what activities are happening during interphase? There is growth 1, synthesis, and G2 or growth 2 phase. So, during what phase of the cell cycle is DNA replicated? So, it's under interphase, specifically synthesis phase or S phase. It's where the DNA is replicated. After G1 yun, or growth 1. Why it is important for the cell to replicate the DNA? As I've said kanina, it should be replicated and distributed correctly before cell division to ensure that the copies of the chromosomes or DNA is transferred from all of the daughter cells. If not, errors could happen. How will you describe the cell cycle using this figure? So, cell cycle is a continuous growth, synthesis of DNA, and cell division. Okay? So, let's watch this video.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into 3 discrete phases called G1, S, and G2. During the G1 or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the two groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of two genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase. And in these phases, duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of two genetically identical daughter cells.
Okay, so now we got an idea about how the cell cycle and cell division works in a cell. Okay, so in this picture, or I mean video, you see that a cell cycle has two parts: the interphase, which the cell only grows and then the DNA replicates and then grow again before cell division. So again, in the cell division, we have prophase, metaphase, anaphase, telophase. Either it is for mitosis or meiosis. Either it is for body cells or mitosis, or for the sex cells or meiosis. Laging may prophase, metaphase, anaphase, telophase. Okay? So, ang pinakang acronym dyan para makabisado yung cell cycle is IPMAT. I, interphase, P, prophase, M, metaphase, A, anaphase, T, telophase. IPMAT. That's the sequence. Okay? So, moving on. So, when we say cell cycle, this is the repeated drama of cell division. So, this is the interphase and the M phase, wherein cell division happens sa M phase. M stands for mitosis or either meiosis. So, after the growth phase or the interphase, kita nyo napakalaki niya. Buong buhay niya halos nag-i-interphase siya. Konti lang yung portion ng M phase or the cell division. And then, after cell division, nagkakaroon na cytokinesis. Bakit ganyan? Kasi itong cell division na to, it is the nucleus that divides first. After ma-divide yung nucleus, after nun, yung cytoplasm na yung nagde-divide. Okay? Kaya, ganun yung nangyayari. Asan dyan sa cell, dito sa picture, yung nucleus, yung violet. And then, yung the blue part, yun yung cytoplasm. So, nauuna mo na ma-divide yung nucleus in a process called karyokinesis bago yung cytoplasm called cytokinesis. Okay? So, interphase muna. Interphase in the cell cycle, ang nakalagay dito, cell is engaged in growth and metabolic process. Yun lang ang ginagawa niya during interphase. Magpalaki na magpalaki na magpalaki. So, growth 1, ayan na nangyayari. Okay? Ma'am, ba't nawala ang ibang parts ng cell? Ayan. Kasi, pinapakita lang muna natin dito yung major parts ng cell and kung ano yung involved lang for cell division and cell cycle. So, makikita natin dito yung cell membrane, nucleus, yung mga chromosomes na nasa loob na maninipis, cytoplasm, yung blue part, and then yung centrioles. Yun yung mahalagang parts dito sa ating cell cycle and cell division. Okay, so growth 1. Ayan lang, nagpapalaki lang siya. After nyan is S phase. nakita nyo, dumoble, kumapal yung mga chromosomes kasi sa loob nun, nandun yung DNA. So, sa S phase or synthesis phase, DNA is replicated. Kailangan yun. Then, pagdating sa G2, mas lalaki pa siya. So, yun yung ibig sabihin ng interphase. Engage in growth and metabolic processes. Okay? Next. Mitotic phase or M phase. Kapag mitosis ang pinag-uusapan or sa body cells, mitosis or mitotic phase. Later, yung meiosis phase ng M phase. Okay? So, ang tinutukoy natin muna dito na parts ng katawan ay lahat maliban sa egg and sperm. Kaya siya mitotic phase. Again, M phase stands for either mitosis or meiosis. So, dahil mitotic phase tayo, ang tinutukoy dito ay body parts maliban sa egg and sperm. Kasi meiosis phase yun later. So, when we say mitotic phase, growth stops. Okay, so, matitigil yung pag-growth kasi sa interphase yun nangyayari. M phase sa tayo, so tigil ang growth. Ito na nangyayari. The nucleus divides first and then the cell divides. Okay, so if the nucleus divides first, that means karyokinesis. Yun yung tawag doon. The nucleus divides. So, asan yun? Ito yun, mitosis phase. Nandyan na si PMAT. PMAT, prophase, metaphase, anaphase, telophase. So, yung nucleus doon, yung una magde-divide before yung cytoplasm. In a process called cytokinesis, pag cytoplasm yung nagde-divide. Let's start with prophase. Ito yung nangyayari sa prophase. Okay? Kanina, maninipis lang yung mga chromosomes. Ngayon, nag-coil na sila. Nag-coil, nag-condense, kumapal, isa lang yung meaning nun. Okay? Next, nakita nyo yung centrioles. Ang nangyayari, dumoble siya para pumunta dun sa opposite pole, sa tigkabilang side. Bakit nandyan si centriole? Kasi tumutulong siya for cell division. Okay? Next. Ayan na. So, ang tawag dyan sa chromosomes na yan, yung magka-partner na yan, ay sister chromatids. Okay? Sister chromatids. Under pa rin ng prophase, makikita natin nawawala si nuclear envelope. So, yung balat ni nucleus nawawala. Nagdi-disintegrate, nagdi-disappear. Okay? So, ganun yung prophase. Next. Si centrioles pupunta na sa opposite side. Then, lumalabas na si spindle fiber. Kasi nga, mag-i-aid na siya for cell division. Next. Ito na yung ending ni prophase. Dapat nandyan na siya. Wala na si nuclear envelope, nag-disintegrate na. Si centrioles ay napunta na. Si centrioles yung mukhang parang chalk dun sa picture. And then, si spindle fiber, yung orange na tutusok mamaya dun sa mga chromosomes. They aid in cell division. Okay, that's their functions. So, yan yung prophase. They prepare themselves for disintegrating the nuclear membrane. Okay, preparing to the centrioles to move to the opposite side. And then, ayan. So, meron din tayong mga coiling or condensing of the chromosomes. Yun yung nangyayari. Next, metaphase. Metaphase na tayo. Meet, align. Ayun, yun yung keyword. Meet, align. So, sa metaphase, makikita natin yung mga sister chromatids. Okay, sister chromatids chromosomes din yung class, pupunta na siya sa center. Ayan. They will meet and align at the center. Yun yung key term. Okay? Metaphase. Metaphase. So, nag-a-align na sila. Nag-meet na sila sa center. Ayan na. Okay? So, si spindle fiber, fiber, tinutusok na niya si chromosomes at the center. Kailangan nasa metaphase plate sila sa equator, sa center. Okay? Naka-align sila properly. So, yung middle section ng ating chromosomes, ang tawag doon ay centromere. So, doon dapat mapupunta yung mga spindle fiber to ensure that they are well aligned or meet at the center. Okay? Next, anaphase. Ana means away. Okay? Kasi magsa-separate sila later. Ayan. Yung sister chromatids natin ay hihilahin ni spindle fiber away from each other. So, si spindle fiber magso-shorten, iiksi. Dahil umiiksi siya, mahihila yung sister chromatids. Okay? So, ano na sila, naghiwalay, they separate. Ana, away. Okay? So, they separate, they move away, they move apart. Yun yung mga key terms sa anaphase. Ayan. So, mas umiiksi si spindle fiber, hinihila niya towards the opposite side, yung mga sister chromatids. Ayan na siya. Then, telophase. Sa telophase, makikita natin, napunta na talaga doon sa opposite poles, no, or opposite sides. Ngayon, magkakaroon ng cleavage or cleavage furrow. Okay? Yung parang uka. Ayan. And then, makikita na natin na yung ating nucleus ay nagsa-start na siyang, nahati na siya. Okay? Mahahati na siya. And then, susunod na doon yung cytoplasm. So, tingnan natin yung susunod na mangyari. Ayan, lumalalim yung cleavage furrow natin. And then, bumabalik na yung ating nuclear envelope na nawala doon sa prophase. Tapos, si chromosomes, okay, na makapal kanina, ay nagiging chromatin na lang. Kasi maninipis na siya. And then, si centrioles ay bumalik na, nawala na yung spindle fiber. And then, ayun na, nakita na natin na nag-divide na fully yung ating nucleus. Ang susunod na dyan is cytokinesis, yung cytoplasm naman. Okay? Ang process kung saan na-divide ang nucleus, asan dyan yung nucleus, ma'am? Yung violet. Ayan, no? Bumalik na nga yung envelope niya eh. Karyokinesis. Karyo means nucleus. And then, yung cytoplasm na yung next. So, after ma-divide ni nucleus, eto na, si cytokinesis. Division of cytoplasm and cell organelles. So, ayan na siya. Lumalalim na yung cleavage furrow, yung cytoplasm na mahihiwa na siya. Ayan na, nagkaroon na tayo ng 2 daughter cells. Okay, so ngayon naintindihan nyo na kung ba't kailangan si DNA during the interphase, specifically S phase or synthesis phase, dapat na duplicate para mabigyan yung daughter cells ng exact copy of the DNA. Ayun yung ibig sabihin niya. Okay? Ayan na. Yan yung overview. Again, cell cycle is a larger portion than cell division. Kasi yung cell division, ito lang yan, oh, yung PMAT, tsaka yung cytokinesis. Yung interphase, growth, synthesis, growth 2. Then, ito lang yung portion ng cell division, onti lang. So, ang key term dyan, para makabisado natin siya, yung sequence ay PMAT, interphase, prophase, metaphase, anaphase, telophase. Gusto mo sama mo na IPMAT C, kasi sa dulo, cytokinesis yung nangyari. Okay? Okay, let's add some more videos.
An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into three distinct phases. During G1 or gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as two sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach, by proteins in their centromeres called kinetochores, to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint, and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.
Ayan. So, narinig natin kung para saan yung mitosis, no? Para siya sa growth, repair, and regeneration ng ating iba't ibang parts ng body maliban sa egg and sperm, no? So I hope na visualize nyo na yung nangyayari sa ating body once na nagmamitosis siya. Okay? So let's answer the activity that you have on your module. So for prophase, the long and thread-like chromatids start to coil. So, nakita nyo naman sa picture, nagko-coil siya, no? Ibig sabihin, nagpupulupot siya, nagko-condense siya, kumakapal siya. The spindle fibers form and attaches to the centromeres, yun yung middle portion ng chromosomes natin. The nuclear membrane and nucleolus disappear. So, nawawala. Disappear, disintegrate yung chromosome. Nuclear envelope. Next, metaphase. Meet. Okay, nag-meet sa gitna. So, it's A. Chromosomes move and align themselves at the center. They meet at the center, metaphase. They align at the center, metaphase. Okay. Next, anaphase, away. So, the sister chromatids of each chromosome divide and split. Divide, split, move away, move to the opposite poles. Yun yung mga keywords. Ba't sila naghihiwalay? Kasi nag-shorten yung spindle fiber. So, hinihila siya to the opposite side. Naghihiwalay sila. And then, may cleavage furrow na during telophase, and this what happens. The chromosomes are located on the opposite poles and the spindle fibers disappears. Ayun, yun yung nangyayari sa ating telophase. The chromosomes uncoil, so numinipis na si chromosome, nagiging chromatin na, and the nucleolus and nuclear membrane begin to form. Bumabalik na ulit yung nuclear membrane na nucleus kasi nag-divide na yung nucleus. Ang susunod naman is the cytoplasm in a process called cytokinesis. Pag nucleus ang nagdi-divide, karyokinesis ang tawag doon. Okay, so why does chromosomes move and align themselves at the center during metaphase? To ensure that the spindle fiber will let the chromosomes meet at the center so that when it divides during anaphase, they have equal number of chromosomes. Remember that chromosomes have the DNA inside, which is very important in our transfer of traits. Okay, kasi nandun yung genes sa loob ng DNA. What happens to the spindle fiber during anaphase? The spindle fibers shortens. Kaya siya naghihiwalay. Okay? Why? Ba't nag-shorten? Para ma-divide yung cell later on. Why mitosis is important? It's for growth, repair, regeneration. Okay? Kasi meron tayong mga parts ng katawan na laging na-burn out, yung tissue, yung cell, na kailangan i-repair. Okay? At ma-reproduce para masustain yung buhay natin. Ayan yung mitosis. Sa interphase, ito ay kunwari ay skin. Katawan ng tao yung tinutukoy, 46 chromosomes. During interphase, ano lang yan? Growth 1, synthesis phase, growth 2. After that, M phase na or mitosis phase, if it is for the body parts. So, yung 46 natin, madidivide siya into two daughter cells which are identical. So, both of the daughter cells will have 46 chromosomes and the other one is 46 chromosomes. Paano naging ganun? Kasi nga, kinapi lang naman niya yung chromosomes. Kaya nga pagdating dun sa interphase, synthesis phase, yung DNA natin ay dapat maduplicate. Para pa naghiwalay siya during cell division, each of the daughter cells will have the same exact copy or number of chromosomes, no? So, kung skin mo yan, ganun pa rin, no? Skin mo pa rin yun. Next, meiosis. So, dito, meiosis 1 and meiosis 2 kasi 2 rounds yan ng cell division. Prophase 1. Prophase, alam mo kahit 1 and 2 yan, na prophase, metaphase, anaphase, telophase, laging yung keyword andun pa rin. Prophase, they prepare themselves na yung chromosomes mo ay mag-coil, yung nuclear membrane ay nawawala, and then yung centrioles ay pumupunta sa opposite pole. Yun, ganun. So, yun yung mga words na dapat tandaan natin para hindi kayo malilito kahit ang dami-dami na nakasulat. So, why letter D? Homologous chromosomes pair by a process called synapsis. Dito sa meiosis, sex cells na yung pinag-uusapan. Hindi na buong katawan, sex cells lang. So, nakikipag-pair si chromosomes, ang tawag doon synapsis. Homologous chromosomes, minsan ba kung pangbuhok na chromosome yun, o sila yung magsasama-sama. Okay? Kung pang skin color na chromosomes, yun yung homologous chromosomes. Okay, so magkakaroon sila ng synapsis, magpapalitan sila ng genetic material, kung kulot ba yung buhok o tuwid, kung brown ba yung buhok or black, ganyan. So, pag nag-exchange sila ng material or traits, ang tawag doon crossing over. So, may occur between homologous chromosomes when the chromatids are broken. Next. So, sa prophase 1 nangyayari ng meiosis lang yung synapsis and crossing over. Prophase 1. Next, metaphase. They meet at the center, they align at the center. Asan yun? Letter A. Paired homologous chromosomes align at the metaphase place. Align. They meet at the metaphase place. The metaphase 1. Next, anaphase. Homologous chromosomes separate. Diba? Alana. Nag-move away, nag-separate. Move toward the opposite pole of the cell by the spindle fiber. Next, Telophase 1 and cytokinesis. Homologous chromosomes reach opposite poles. So, nakarating na siya dun sa tigkabilang side. Then, magkakaroon ng cleavage furrow and then cytokinesis na, 2 daughter cells na. Pero meiosis to. So, may another round para maging 4 na daughter cells. So, prophase 2. Uulit ka sa prophase 2. Prophase 2, spindle fibers form again. So, kung ano yung nangyari sa prophase 1, ganun din mangyari sa prophase 2. The chromosomes condense once more. So, ganun, di ba? Sabi ko sa'yo, pag prophase, nagko-condense yung chromosomes, nawawala yung nuclear envelope, ayun, pupunta si centrioles sa tikabilang poles, then may spindle fiber. Prophase na prophase, di ba? Pero this time, dahil pangalawa na siya, prophase 2. Next, metaphase. Magmi-meet ulit sila sa gitna. So, the chromosomes align at the metaphase play and are attached their centromeres to the spindle fibers. Next, anaphase, maghihiwalay ito ulit. The chromatids move to the opposite poles. This time, yung anaphase 2 versus sa anaphase 1. Sa anaphase 1, may partner pa. Nakikita nyo sa number 3. Sa number 7, na siya? Tuluyan na talaga silang magkakahiwalay ng mga sister chromatids niya. Single na lang sila. Okay? So, next, telophase 2 and cytokinesis. Nuclear membrane is formed. So, bumabalik na, no? Kasi nagka-karyokinesis na siya. And then, the nuclear membrane is formed around each set of chromosomes and spindle fibers disintegrate. Nawawala na si spindle fiber. Nagre-reappear na yung nucleus and the nuclear envelope. And then, after nun, cytokinesis na. So, yung unang daughter cell nag-divide into 2, then the second daughter cell nag-divide into 2, forming 4 daughter cells, which are haploid. So, ang chromosomes nito ay hindi na 46 tulad ng mitosis. Ito ay haploid na. Haploid means 23 chromosomes kung human. Sa mitosis kasi diploid. Okay? Kung 46 yung isa, 46 din yung isa. At identical, pare-parehas. Okay? Ito hindi. Magkakaiba. Tingnan nyo, magkakamix na yung color nila. Okay? Nung chromosomes. Kasi ano na siya? Magkakaiba. Okay? So, yan yung meiosis. Ganun pa rin. May interphase pa din, tsaka M-phase. M-phase this time for sex cells are called meiosis. Meiosis phase. So, tingnan nyo dito. 46 sa una. Then, pagdating sa meiosis 1, nagkaroon ng 2 daughter cells, nagkaroon 23 chromosomes. Pero dahil may second round of meiosis 2, mahati ulit yung tigisang daughter cells into 2. So, forming 4. So, 23 chromosomes pa rin silang lahat. And at the same time, single na lang yung mga nila. Wala nang pair. Yung tawag doon chromatids. Okay? Hindi na sila sister chromatids. Chromatid na lang. Okay? At magkakaiba ang color nila, which means hindi sila identical. Variety sila. Iba-iba. Okay? Next naman. Okay, let's watch this.
Have you ever wondered why children do not look exactly like their parents? Or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis 1 and meiosis 2. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase 1 of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase 1, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase 1 concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers forming the meiotic spindle. In prometaphase 1, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase 1, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles. Meiosis 1 ends with telophase 1, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the two daughter cells are separated by a cleavage furrow. Since the final product of meiosis 1 is two haploid cells, meiosis 2 begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs, and cleavage furrows separate the two daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.
Ayan. So that explains the process of meiosis and its role in the passing of the traits na unique and distinct from the parents. Kaya napakahalaga ng meiosis sa atin in terms of the passing of characteristic para magkaroon tayo ng diversity na tinatawag. Pero mahalaga din yung mitosis kasi nga is for growth, repair, regeneration. Yung meiosis naman for reproduction. Okay? So, haploid, ang nangyayari sa meiosis, say for example, 46 ang chromosomes ng buong katawan, pagdating ng egg and sperm, 23 na lang sila. Kasi haploid. Diploid naman pag mitosis kasi identical, 46 yung isa, 46 din yung isang daughter cell. Okay, that happens for all parts of the cell, body cells, except egg and sperm. And they are identical. Okay? So, move over lang tayo dun sa pictures, dun sa videos. Ito yung nakikita nating the blue ones are the interphase sa cell cycle. And then, yung mga broken arrows from meiosis to cytokinesis are the M phase. So, meiosis siya, M-phase na meiosis, it means nangyayari siya sa egg or sperm. Okay? So, 2 rounds ng cell division na nangyayari sa meiosis. Okay, meiosis 1, meiosis 2. 2 rounds na cytokinesis, kaya nagkaroon ng 4 daughter cells. So, pakilala natin, no? Yung violet na nakita nyo, are X chromosomes. Okay? Ano yung itsura ng Y chromosomes? Itsurang Y. Okay? Itsurang Y, yung ano niya, yung itsura kaya Y chromosomes. Pero apat pa din yung kamay at paa nila, ha? So, sister chromatids, yung tawag dun sa dalawang magka-partner na chromosome, centromere yung nasa center nila, kinetochore yung dalawang proteins na nasa tigkabilang side. And then, si centrioles yung color brown, then spindle fiber yung mga patusok-tusok na nagra-radiate. And then, yung nasa loob ng nucleus, yung mga chromosomes. Okay? Ayan. Sa prophase 1, meiosis 1 tayo, ha. Nagkakaroon ng synapsis, yung pagpapatong or pairing. Doon siya nangyari. Then, crossing over kapag nagpalitan ng traits. Ayan. So, nag-synapsis na siya, nagpatong na sila, or synapsis, then magpapalitan. Ayan, o. Yung blue na halo dun sa violet, yung violet na halo sa blue. Ang tawag doon, crossing over. Pagpapalitan ng traits. Okay? Para pa nag-iwalay sila, iba-iba na yung mga cells. May iba't iba na silang traits. So, nawawala yung nuclear envelope ng nucleus during prophase. Si centrioles, ayan si centrioles, napunta na siya dun sa tigkabilang side, si spindle fiber, lumalabas na. Ayan. Ito yung sa mitosis. Ganyan lang sa mitosis. Pagdating sa meiosis, may synapsis and crossing over. Yun yung pinagkaiba na mitosis and meiosis. Kineto ko yung tawag dun sa dalawang protein na nasa tigkabilang side ng chromosomes. Okay? Ito na yung metaphase. Nag-meet na sila sa center. Okay? Tinusok na sila ng spindle fiber sa kanilang centromere. And then, anaphase, away. Okay? Meta, meet. Meet, align. Ito naman, away. Okay? Naghiwalay sila during anaphase. Pero tinan nyo, magkakaiba na yung characteristic nila. Iba-iba ng color. And then, pagdating sa telophase, nagkakaroon ng cleavage furrow dun sa cytoplasm. Hihilayin na siya ng hihilayin hanggang sa mag-divide yung nucleus. Ang tawag karyokinesis. And then, ang susunod na ay yung cytoplasm. Ayan, magsa-cytokinesis na yan, mapuputol na. So, cleavage furrow yung parang deep na na nakikita natin na puputol dun sa cytoplasm. So, the cytokinesis, it's the cytoplasm that divides. So, mauuna muna ang karyokinesis or the nucleus that divides before the cytoplasm divides. Cytokinesis naman yun. So, another round. Meiosis 2 na tayo. Uulit lang ulit. So, si prophase, mawawala yung nuclear envelope. Si centrioles, pupunta sa tigkabilang poles. Ganun ulit. Okay? Then, si spindle fiber, lalabas. Pagdating sa metaphase, magmimit sa center. Ayan, mag-a-align sila. Ayan yung metaphase plate natin. Anaphase, ganun ulit. They move away. Ayan. This time, single na silang dalawa, yung chromatids. Ayan, magkakaroon ng cleavage furrow. Then, telophase na-divide na, nag-karyokinesis na yung nucleus, nag-divide na. Susunod na si cytokinesis, yung cytoplasm. Ayan, nag-divide, nakita nyo, nag-divide na yung nucleus, na siya, nagiging visible na yung nuclear envelope. Bumabalik na yung nucleus. Eto na. So, cleavage furrow deepens, then cytokinesis, ayan na, mapuputol na yung cytoplasm. For daughter cells na magkakaiba na ang traits. Ayan na. So, yung apat na yon ay in a gamete formation or sexual formation, it can either be egg and sperm. Okay, so nakita natin, kapag buong part ng katawan, mitosis, diploid yung tawag doon. 2N, 2 times N. N means 23, isang set ng chromosome yun. 2 times 3, 46. So, 46 chromosomes ang lahat ng nasa katawan natin, maliban sa egg and sperm. Okay? So, may meiosis yung ating cell. Ito naman ay N na lang, kasi haploid. So, si 46, magiging isang set na lang, 23. Okay? So, from 2N naging N. N means 23, isang set. Diploid, meaning half. Half ng 46. Okay? Ayan. So, si haploid ay either si sperm or si egg. Bakit 23? Kasi pag nag-fertilize sila, na-fertilize ni sperm si egg, 23, 46, mabubuo ulit yung baby. Pero pagdating dun sa egg and sperm niya, kung babae man yan o lalaki, 23 lang. Para pag may naka-partner ulit siya, mabubuo ulit yung 46 baby ulit. Diba? Yun yung essence niya for reproduction. So, difference between mitosis and meiosis, ayan, 2 daughter cells sa mitosis. Okay? 4 sa meiosis. Yung chromosome sa mitosis ay hindi nahati, kaya diploid ang tawag, pero sa meiosis, haploid. Pagdating sa mitosis, walang homologous chromosomes na nagpe-pair. Pero sa meiosis, yes. Sa prophase 1, nandun si synapsis, yung pagpapatong ng homologous chromosomes, pairing, tas crossing over o palitan ng traits para maging magkakaiba sila later on. So, which means mitosis, identical yung nabubuong daughter cells. Pero pagdating sa meiosis, may variation. Okay? Ano yung no dyan? No, hindi siya identical kasi variety, iba-iba. Isang beses na nagkakaroon ng cell division sa mitosis, 2 beses sa meiosis. Meiosis 1, meiosis 2. So, 2 rounds ng prophase, metaphase, anaphase, telophase. Ang chromosome number na meron sa daughter cells ng mitosis ay 46. Sa meiosis naman, lahat silang 4, 23. Ang formula ng mitosis 2n, so 2 times 23 is 46. Kung yung chromosomes ay pantao, 46 tayo. Meiosis naman n lang, which means isang set ng chromosomes, 23 yung equivalent. Okay? And then, let's answer the spermatogenesis, formation ng sperm sa ating mga sex organ. Oogenesis, formation ng egg cells sa ating female reproductive organ. So, ito yung mga spermatogenesis. Ayan, nag-o-occur sa testes. Motile gametes meaning gumagalaw kasi may buntot yan, di ba? Yung sperm, flagella. Spermatocytes is produced throughout lifetime of the male. Pag oogenesis naman, sa ovary naman yung nangyayari sa mga babae. It give rise to 2 cells of unequal sizes. Makikita nyo later kung anong tawag doon. Sa 4 na nabuo, yung isa lang doon yung malaki, then 3 puro maliliit. Next, it produces non-motile. Ibig sabihin, walang buntot, di gumagalaw. Then, ito na yung both. Oogenesis and spermatogenesis begins during puberty stage. Give rise to 4 haploid cells. Lahat naman sila 4. And then, begins during fetal development. Okay? So, when we say gametogenesis, gametes means sex cells, genesis means beginning. Okay? So, it means gamete formation where daughter cells or gametes are produced at the end of meiosis 2, resulting in the production of egg and sperm. So, pag nagkaroon na ng 4 na daughter cells after meiosis 2, magsa-specialize na siya, either magiging sperm yun or egg yun. Okay, so, yun yung end point niya. So, gametogenesis, which means general term siya for the formation of sex cells, whether egg or ovum in another term, and sperm, which means yung sa lalaki. Okay? So, ito yung makikita natin. Same yung nangyayari na process sa formation ng gametes both in female and male. It all starts with the interphase. So, mag-grow lang siya. Okay? Padadamihin lang niya identically. Pero after the growth, mag-i-stop yon, magkakaroon ng M phase. Dahil egg or sperm ang mangyayari, meiosis siya. Hindi mitosis. So, it starts with spermatogonium or oogonium. Laging may gonium in the beginning. After that, followed by oocyte. Is either spermatocyte or oocyte. Sperma kapag sperm. Oocyte kapag ovum or egg. Okay? So, primary spermatocyte pag sa lalaki, sa babae, oocyte, primary oocyte. And then, magkakaroon ng unang round na meiosis. So, prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis. Ayan. Nag-form sila ng 2 daughter cells. Ang tawag doon sa nabuong 2 daughter cells ay secondary spermatocyte. Sa kabila, ganun din. May site din. Secondary oocyte. O means ovum. Sperma means sperm. Okay? Pero tingnan nyo yung nangyari, oh. Yung 2 daughter cells dito sa female, sa secondary oocyte, isa maliit, isa malaki. Ganun talaga, unequal size siya. Okay, that's its nature. Ang tawag dun sa maliit ay first polar body. And then the first one, yun lang yung secondary oocyte. And then, second round ng meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis 2. So, yung sister chromatids, maghihiwalay na sila talaga. So, magkakaroon siya ng yung unang spermatocyte, secondary spermatocyte, nagkaroon ng 2 daughter cells. Yung pangalawa, nagkaroon ng 2 daughter cells. Ang tawag dyan, spermatids. Doon naman sa kabilang side, sa babae, yung secondary oocyte and first polar body, 2 rounds ulit ng mitosis, forming 4 daughter cells. 2 doon sa kabila, 2 sa kabila. Again, isa lang yung nagiging malaki. Okay, the rest, maliliit. So, this time, ootids. Ootid. Then, yung isang, daughter cell, polar bodies. This time, second polar body kasi may first na. Yung anak ng first polar bodies din. Okay? And then, magkakaroon na siya ng differentiation. Yung spermatids, magde-develop ng flagellum. Pag marami flagella, buntot. So, motile sila, gumagalaw. Pero yung ovum, non-motile, wala silang buntot. Saan na napunta yung mga polar bodies? Mawawala yan. Okay, mawawala. Kaya mas mabilis mag-menopause ang babae kasi konti lang yung ovum or egg cell na napoproduce kesa dun sa million of sperms na mga lalaki. Okay? Paano nyo ito mga kabisado? Anong technique? Tandaan nyo lang to. Gonium, site-sited, Ulit. Gonium, site-sited, Pag sperm, lagyan nyo lang ng sperm. Spermatogonium. Next, primary spermatocyte. Next, secondary spermatocyte. Then, spermatid. Then, sperm. O, diba? Tama? Gonium, site-sited, Sa kabila, ganun din. Dagdagan nyo lang ng O. O, gonium. Then, the primary oocyte. Then, secondary oocyte. Then, ootid. Then, ovum. Gonium, site-sited, O, diba? Yun yung technique para makabisado nyo yung sunod-sunod niya. Well, anyway, spermatogenesis is the formation of sperm cells, while oogenesis is the formation of ovum or egg cells. Ayan, primary oocyte sa oogenesis, 2N pa siya, pero pagdating doon sa second round na meiosis, na nagkaroon na siya ng cell division, or 2 daughter cells sa secondary oocyte, na yan, haploid na. Okay, haploid. Lahat yan, kahit polar bodies and ovum. Okay. At ganun din sa spermatogenesis, no? 2N pa sa primary spermatocytes, pero pag nagkaroon na siya ng cytokinesis 1, nag-divide na siya, N na lang, haploid na. Hanggang dumating na yung last stage ng cytokinesis, nag-specialize na yung mga spermatid, nagkaroon ng buntot, naging sperm na. Okay? Ganun siya. And then, ito na yung ating activity sa picture sequencing. Ayan. Dito, sa wound na to, nagmamitosis yan kasi hindi naman yan egg and sperm So, mauuna dyan yung hemostasis, nagdurugo, followed by mamamaga, inflammation. Sunod, proliferation, yung magkakaroon na ng mga nana, tapos unti-unting matutuyo. And then, remodeling, yung mga collagen fiber, magbibigay na siya ng new skin. So, ganun yung process ng mitosis, no, sa skin na regeneration. Kasi nga siya ay growth, repair, regeneration. So, sagutan nyo lang tong reflective question na to, process ng wound healing, paano mo explain yung relation dun sa mitosis. Okay? Ayan yung pagkasunod-sunod niyan. So, thank you so much for listening. I hope you learned something. If you have questions, do not hesitate to chat your teacher on the GC. Okay? Have a nice day and goodbye.